the syllabus point that we're talking about today is syllabus point 74, which is to do with the mechanisms of evolution. So we've discussed the phylogeny and evolution in general with all the different types of species and all the different organ systems. But how does evolution actually happen? So it's thought to happen in two ways. The first is through anagenesis, and anagenesis is basically the genetical changes which happen within a species. But these changes are kind of small, they're not like big changes. They're just small changes which happen within one species, and then this doesn't like this these changes are not big enough to allow the species to become different species, to branch off into different species. By the way, can you see the screen, the uh, the slide? Okay, good. All right. Then the second level of evolution is cladogenesis. This is when these species accumulate so many genetical changes that they actually branch off into different species. So that is cladogenesis. So when we're talking about the mechanism of evolution, not just for this syllabus point 74, but also for syllabus point 75, you should know that anagenesis and cladogenesis go hand in hand. They are a combined they're a combined thing. So anagenesis happens first, then we go into cladogenesis. Now, what are the evolutionary steps that happen? So, generation of diversity. So, diversity happens through mutations and genetic recombination. This syllabus point actually relates to, I think it was syllabus point 25 or 24 also, which also talks about differences within species that can happen as a result of natural selection or genetic drift. Do you know what natural selection is? Could you explain what natural selection is? Adnan? Okay.
So it was introduced by Charles Darwin. It's to see which environment the organism
Yes,
is best adapted to live in or survive in or produce.
exactly.
Yeah.
Exactly. So the organisms basically, it could be that we have two populations or two organisms, or one population of organisms, and it could be that there could be some level of something would change. For example, you could have giraffes, right, with long necks, and you know these giraffes, their primary diet could be the trees, the leaves on the tree branches, okay? And so what could happen is that many of them could be eating off these same trees, and then maybe the tree isn't like producing as many leaves as, quickly as possible. So what would then happen? Well, the population would decrease in size because not all of them could get that food source. So a random mutation could happen where some of these giraffes will now develop longer necks, and now they'd be able to get the leaves which are higher up in the tree, for example. So that is an example of natural selection. Now, genetic drift is something that is also covered in the earlier syllabus points. As I said, I think it's either 24 or 25, either 23, 24, or 25 of the syllabus. And with genetic drift, that basically is the change in allele frequencies. So the alleles, basically the frequency of the alleles could either decrease or increase due to a change that would happen within that species situation or environment. For example, bottlenecking events. Do you remember what bottlenecking is? Or do you know what bottlenecking is?
Yeah. So bottlenecking is when there's a population,
Mm,
and because of like maybe a circumstance or like a disease, let's say,
mm,
disease breaks out and it reduces the number of the in the population, and the
exactly.
ones that do survive maybe have the genes to survive the disease and so on.
Yeah.
And yeah,
Exactly, exactly. So, a very good example of what would cause genetic drift would be a bottlenecking event. So, exactly as you described, a group of species may be exposed to a disease or maybe exposed to like a geographical change or something that would split them up from the rest of the population. This would mean that the surviving population, as you said, may have the genes which allow them to survive. That original bottlenecking event, and because they survived, they are then going to be able to pass on their surviving genes. Okay, so cladogenesis we described very briefly, but basically it is when a species is basically able to split up into two or more different species. Okay, and then what will happen is that it will cause these species to now be able to develop independently. Okay, and we are going to talk a little bit more about that in the next syllabus point. So now, we mentioned that mutations are, of course, a common reason for why evolution would happen. So most of these mutations are usually point mutations, and these point mutations are mutations that affect a single nucleotide within the DNA sequence. This syllabus point, so this part about point mutations and evolution, also ties in with the earlier syllabus points of biology, um, because you do have to know what substitution, insertion, and deletion is. But we are going to go through that together now. So just bear with me. Let's zoom in a bit. Okay, you can still see the screen, right?
Yep.
Okay, cool. So this is our DNA sequence, of course, at the top, and then here is our mRNA sequence too, which corresponds to our DNA sequence, right? As we know, we need this mRNA, this template, in order to make the proteins eventually. We know that proteins are made out of amino acids. Okay, so here we have phenylalanine, tyrosine, glutamic acid, for example. Okay, so this is in a normal situation. We have normal DNA, a normal sequence of DNA nucleotides, and then we have a normal sequence of mRNA nucleotides that are corresponding. Okay, now in a substitution, what would happen is, of course, exactly as the name suggests. So the nucleotide would be substituted and replaced. So another nucleotide would come into that sequence. And this would happen right here. So let's say here we could see that the DNA in this section right here, we've got cytosine, right, and then thymine and thymine. But here we have thymine instead of cytosine. This means that the mRNA molecule, which is of course supposed to be copied complementarily to the DNA, instead of it forming a or having a guanine nucleotide base here, it's going to have a adenine nucleotide base here instead, right? So, in order to match in with that thymine. So, what's going to happen is this is now going to be a completely new nucleotide sequence, and this is now going to code for a completely different amino acid. So, instead of coding for the glutamic acid, which is what we needed, we are now going to make lysine. So, that is a substitution. Then we have insertion. So, again, insertion is basically as the name suggests. So, another nucleotide base is going to be inserted into the DNA sequence. Okay, so the original here was CTT, but what's happening here is we've added another C. Okay, so this is shifted, right? This is why we can also call this a frame shift mutation, because there's a shift happening, which is going to affect the next nucleotide sequences. So now we were supposed to have CTT, now we have CCT. And where did the other T go from previously? It went over to the next sequence, right? Which means that now we're going to make two completely different amino acids than what we started off, or what we wanted originally. Okay, which we can see right here. So that's insertion. And then deletion, again, as you can see in the name, it basically just means that we are deleting a nucleotide base. Again, this causes a shift. So then we get 3 new amino acids. Okay, and this will, of course, result in a completely different protein that is going to be made. And, of course, this could then either positively affect our evolution or negatively affect our evolution. Okay, because it could either cause proteins which are functional or proteins which are not really functional. But usually, missense mutations, this is where there is a change in one amino acid and it becomes another amino acid, for example, in the substitution. This could sometimes be beneficial. But then, if it's a nonsense or frame shift mutation, like in insertion and deletion, this could cause a completely different protein to be formed, potentially a non functional protein. Okay, so an example that they mention in the lecture slides to do with point mutations and when these point mutations could be beneficial is in the case of antibiotic resistance in bacteria. Of course, this is not something that is good for us, but it is something that is good for the bacteria. So, systemic use of antibiotics, if we're constantly using antibiotics more and more and more and more, this is going to lead to the bacteria which we're using these antibiotics against to develop some type of resistance. And they're able to develop this resistance through these random point mutations that they adapt in order to, of course, adapt to the antibiotics and to overcome these antibiotics. And how do they pass these point mutations on? Well, they have these structures called plasmids. Okay, so within their plasmids, they can basically pass on this antibiotic resistance gene from one bacteria to another. And this is something that I will discuss later here. But an example that the lecture slides also mentions is in the example of the TEM1 beta lactamase. So, mutations within the TEM1 beta lactamase enzyme basically allows for there to be resistance to this antibiotic called cephalosporin. You don't really need to know in detail exactly how or what's exactly going on, but this is just an example of a type of bacteria that was able to develop a resistance to a specific type of antibiotic. Okay? Different mutations, of course, can lead to varying degrees of antibiotic resistance. Some mutate, but then it's all very random. It's all very random. This is another image that was also within the lecture slides. And this basically just shows you which antibiotics are used most frequently in which areas of the world. And of course, wherever those what's it called? What was I going to say? Wherever they use the most amount of these specific antibiotics is wherever they have to be the most careful when, of course, prescribing these antibiotics. Because if they use like a lot of, for example, clarithromycin, as we can see, it's 92.4% here within Africa, they have to kind of be careful to not administer as many, because the bacteria that they're trying to fight against may have developed some level of resistance because of these point mutations. Okay, so gene duplication is another thing which we have to know and discuss. So gene duplication is, of course, the duplication or the doubling up of a gene. And this could happen in crossing over, in recombinational repair, or in site-specific recombination. And this basically brings exons from different genes together. Okay, now what are the exons? Do you remember? What do the exons do within the gene?
So the exons, they
Mm,
so in the gene itself, there's
yeah.
for the sequence for the mRNA,
Hmm.
they they code for the protein, and
Yes,
yeah,
exactly.
so basically, yeah, they code for the protein and are responsible for any transcription. For example,
Mhm.
both exons and introns are responsible for the transcription of the mRDNA, mRNA. I mean.
Okay, so so the exons are the coding portions. Then the introns are there for gene regulation. But of course, as you mentioned within the mRNA, when we want to make mature mRNA, we remove the introns. We're only left with the exons, which are the coding parts, right? And what removes the introns off of the mRNA?
So the introns,
Yeah,
mm,
so they're removed by something. In the process of mRNA maturation, when we want to make mature mRNA and use that for our transcription, we remove the introns, but the introns are removed by a specific thing. that you have to know the name of?
I don't remember, to be honest.
Okay, no worries. The spliceosome. Okay,
Yeah.
so the spliceosome is what actually removes the introns away from the mRNA. Okay, so now these duplicated genes are able to evolve new functions and new gene families. Of course, if we have more genes, that means that we're going to make more and new combinations of proteins, right? And an example of this is the sRGAP2 gene, which is basically found within. It's basically found to influence the development of the cortex within the brain. Now, humans are unique as we have multiple copies of this sRGAP2 gene. Okay, we have multiple copies. We can see in this image we have about four. All right, whereas other mammals they may only have one sRGAP2 gene, and it's basically found that these sRGAP2 gene may have a role in enhancing our neuronal connection, as they may have a function where they could maybe form what's it called more dendritic, basically dendritic fibers within the neuron. Okay, this is of course something that's still being researched, but as we mentioned, humans have duplicated sRGAP2 gene. So in some cases, this gene duplication, which is something that happens over evolution, is a necessary and positive thing. Okay. Okay, so in this slide, we have to learn about the how do we pass on these beneficial mutations. So in eukaryotes, there's two mechanisms. There's sexual reproduction and genome cleansing. In sexual reproduction, obviously that is very straightforward. We are able to pass on our genetic information to our offspring, and this promotes genetic diversity, of course. In genome cleansing, we're able to remove and delete our deleterious mutations. These are mutations which result in non-functional proteins. So we're able to just get rid of these non-functional proteins within the meiosis and fertilization. Then in prokaryotes they use horizontal gene transfer. Horizontal gene transfer is something which we've discussed before, and we spoke about this in syllabus point 60. Yes, and basically it is different to the vertical gene transfer. So the vertical gene transfer is what we all know. Okay, vertical gene transfer is basically that gene transfer that happens between parents to offspring. And horizontal gene transfer is basically the genetic transfer that happens between one species to another species. And this can happen through transformation, transduction, or conjugation. Okay, and this is usually, of course, non-sexual. And this is able to spread beneficial mutations like antibiotic resistance across different bacterial species. Now we're going to look into these horizontal gene transfers. So, in bacterial transformation, the donor cell, the donor bacteria, might sort of just release its DNA into the surrounding environment. Okay? As part of its DNA, it may have the antibiotic resistance gene that's basically attached to it, and then it would just release it into the environment. Then there will be a recipient cell that's just there minding its business, and it will actually take in this DNA from the environment. And this is how this bacteria, the recipient cell, was able to get the antibiotic resistance gene from the donor cell. That is bacterial transformation. Then we have bacterial transduction, and this is normally something that involves a bacteriophage. Do you know what a bacteria? Whoa, sorry, sorry. Do you know what a bacteriophage is? A
No.
bacteriophage.
Uh,
Okay,
yeah. Go ahead.
bacteria. Yeah, sorry, go on.
It's okay. I was gonna say, no problem. Whatever problem happens, I will just edit out anyways.
Okay, inshallah. Thank you. Do you know what a bacteriophage is?
A bacteriophage?
Yeah.
No.
Okay, this is a bacteriophage, and essentially, what it is just these viruses which specifically infect bacteria. As we know, viruses are not able to sort of live on their own and reproduce on their own, so they have to infect bacteria. They have to go or infect other host cells. But bacteriophages, such as this one, specifically only infect bacteria. So a bacteriophage will come along and it will basically go into the donor cell, and it may take up some of that DNA of the donor cell. It may take up the antibiotic resistance gene, and then it will leave the cell, the donor cell, and then it will go into the recipient cell, and it will attempt to infect the recipient cell with its own genetic information, or it will inject the genetic information which it took from the donor cell. And in this case, of course, it will also give the recipient cell this antibiotic resistance gene. Okay, so that is bacterial transduction. Then we have bacterial conjugation, and this is basically when two bacterial cells, they come together, they basically end up sharing their cytoplasm. They merge together, fuse together, share a cytoplasm, and through this, they're able to transfer their plasmids. Remember, I said what plasmids were before, but plasmids are these vectors where they are able to place part of their genetic information into them, and then they're able to pass this plasmid from one donor cell to the recipient cell. Okay, so this is how they're able to pass on their antibiotic resistance from one donor cell, from one bacterial species to another bacterial species. Then we've got polyploidization. So this is whole genome duplication. Whole genome duplication is very, very important. It's also something that we discussed earlier in syllabus point 61. And whole genome duplication, if you remember, is what was responsible for the formation of tetrapods. Do you know what a tetrapod is?
Yeah. So a tetrapod is a basically an animal with four
Yes,
limbs, basically.
with four limbs, exactly. So we were able to go from, when basically in the formation of vertebrates, we were able to go from animals which didn't really have any limbs at all or even fins. For example, the agnata, right, which only has the caudal fin, to animals which then later on developed four limbs, right? So this happened as a result of the whole genome duplication. So this is a duplication of the entire genome, which is a set of genes, and this results in tetraploidy. Tetraploidy is having four sets of a chromosome, okay? Now, this basically provides new genetic material for mutations and selections because, of course, now that we have more chromosomes, of course, we're going to have more genes. And if we have more genes, that means that we're going to have more proteins. Okay, so new genetic material for mutation and for selection. And if there were any mutations existing in the previous singular chromosome, this is now going to be duplicated when we end up with what's it called, four extra chromosomes, or not four extra chromosomes, but what's it called, the doubling. Okay, this is going to lead to more of these duplicated genes, and we could have more functions. Now, tetraploidy, okay, is more what's it called? It's something that usually happens more within plants. For example, in the lecture slides, they mention wheat. Okay, and in these plants, it basically allows these plants to, for example, develop basically nicer tasting nectar for example, the bees to come and to collect the nectar at the same time for them to allow the plants to, what's it called, spread the pollen around. Um, and yes, so basically, this tetraploidy or polyploidization is something that is very beneficial for the plants. It was also beneficial for us before, as we mentioned, as it happened two times. And as we mentioned, allowed for basically the vertebrates to go from having no limbs to then having four limbs. So it allowed for some complexity to happen, for us to become more complex and for us to be able to adapt to our environment better. Okay, so the gradual evolution of complex structures. So what you need to remember when it comes to evolution is that all of these changes are changes which happen slowly over time, then they're not things that happen straight away and very quickly. They're not big changes, they're small incremental changes. Okay, and scientists are able to research the evolution by looking at fossil records, and through these fossil records they can see these gradual changes as these animals adapted to from living in life in water in aquatic environments to living on land. Because as we mentioned before, we said that life started with the what's it called, the bacteria, of course, and then this, or the RNA, which then formed prokaryotes, for example, like bacteria. Then we mentioned how we got later on, this became more complex as oxygen was introduced into the air. And then we also mentioned how these prokaryotes became multicellular eukaryotes over time, right? That were still living in the water. For example, the porifera, which are the sea sponges, right? Then over time, they then developed limbs and other structures which they would need in order to adapt to life on land, right? So, the evolution of complex structures like the eye, for example, can very easily be seen in the group of animals called mollusks, right? So, because of that, mollusks are very heavily researched within evolution. Because remember, we mentioned the pinhole eyes that they have, right? So, one group of species of mollusks may have pinholes, and then you could see in other animal groups, of course, we developed from that, and then we developed further more complex eyes. Okay, do you have any questions? Because we are at the end of the syllabus point.
No questions.
Okay. Now we are going to go to the next syllabus point, which is 75. Okay. So we basically discussed the first mechanism of evolution. Now we're talking about the second mechanism of evolution, which is when these animals are dividing into different groups or different species. Okay, so what classifies a species as a species? Or so, for example, let's say you have like one group of animals and then they've developed all of these mutations over time. What is going to separate them into two different species? Well, what's going to separate them is the development of reproductive isolation mechanisms. What does this mean? Okay, this basically means that these animals that used to belong to the same group, okay, they've become so genetically different that if they tried to reproduce with one another, that wouldn't be possible. They wouldn't be able to produce fertile offspring. Okay, so that is what basically classifies two different species as two different species. Okay. Now, these reproductive isolation mechanisms, there's two that you need to know. There's the prezygotic isolation and the postzygotic isolation. In the prezygotic isolation, these are the mechanisms that will stop the zygote from forming in the first place. As we know, the zygote is the combination of the sperm cell and the egg cell. So in prezygotic isolation, the zygote will not form at all. In post-zygotic isolation, the zygote will form, but there will be further problems later on. Okay, so what will cause the zygote not to form at all? There could be habitat isolation, as these two populations, they may be living in completely different environments, and because of this, they won't see each other at all, so there won't be any chance of that reproduction to happen. There could be temporal isolation, so this could mean that the populations reproduce at different times, and because of this, they wouldn't be compatible. There could be behavioral isolation, so differences in mating behaviors. There could be mechanical isolation. It could be that their reproductive organs are not compatible, and so, of course, that wouldn't work. Or there could be gametic isolation, which would mean that the sperm and the egg would not be able to fuse together. So these are prezygotic isolations. Now, in postzygotic isolation, where the zygote is actually formed, what happens is once the zygote is formed, so they basically have the baby. It could be that this hybrid that is formed between these two different species may not be able to develop, and they may die early. So this is called hybrid inviability. Okay, or it could be that their hybrid, their offspring, is surviving. Okay, but they are sterile, meaning that they are not able to basically reproduce. Okay, so they are not able to mate and then reproduce. So an example of this would be mules. Mules are basically the hybrid between a horse and a donkey. So when a horse and a donkey sexually reproduce, they will form a mule. These mules are not able to sexually reproduce with horses or donkeys or other mules, and they're not able to basically what's it called, form a offspring. So this is hybrid sterility. Then there's hybrid breakdown. So this would mean when the hybrids are fertile, so they're not sterile, they are fertile, but their own offspring, so after that, so when they have their own offspring may be inviable, so their own offspring may die early or may become sterile. Okay, so these are examples of what could happen with post-zygotic isolation. And if you remember, we said these species are classed as different species if there is reproductive isolation mechanisms in place, meaning that these two different species are not able to produce fertile offspring or offspring that are able to thrive, offspring that are able to live. So, stages of speciation. So, first, of course, there will be the initial divergence where there will be genetic differences, so many genetic differences which will happen in the isolated populations because of mutation, selection, or genetic drift. Okay, and then there could be the post-zygotic isolation or the pre-zygotic isolation, as we mentioned before. So, this is an example of geographical isolation. So, we could have two populations that are basically of the same species and they're living in the same sort of environment. Then, a physical barrier will come in the middle. For example, mountains may just form in the middle, and this will then separate the species. You know, the two, I mean, they weren't two separate groups, they were the same groups, but now they've been split up into two different groups as they're living in two different sides of the mountain. Now, these two different sides of the mountain may have different climates. For example, it may be more sunny in one area, may be more rainy and cloudy in the other. And because of this, these two different groups are now having to adapt to the different environments or situations that are happening around them. Okay, and because of this, they may develop new features. Okay, because they will have these genetic mutations that we spoke about before, and what's it called? And this will basically allow them to form and basically become different species over time. So, what are the types of speciation? So, this is important to know. We have allopatric speciation and we have sympatric speciation. Allopatric speciation is in the example, basically just as the example that I explained earlier, which happens when these populations are split due to geographic reasons. Okay, for example, mountains may form in the middle or rivers may form in the middle. And an example is the Darwin's finches. So these are the birds which were living in the Galapagos Islands. Okay, so they developed basically different beaks, which we're going to talk about later, but they developed different beaks as they were living in different islands and they had different climates. And they needed these different beaks as they were eating different things. So some of these finches, they may have been eating tree branches, so they'd need to have maybe stronger beaks, and then some of them may be eating insects, so they would need beaks that are grasping onto things easier. Or some of these birds, these finches, they would need to be eating seeds, right? As maybe that was all that they had available, and because of that, their beaks would of course need to be stronger in order to crack open those beaks. Okay, so this is an example of allopatric speciation. Sympatric speciation happens without geographic isolation and is usually a behavioral thing. Okay, for example, the chichled fish, which you can find in the African crater lakes, they were swimming in these lakes, right? as a group, everything was fine and normal. But then eventually, some of these chilled fish decided to venture further out into the lakes or different areas of the lakes, okay, where that environment may have been different. And again, this was a behavioral thing. It wasn't that it was a geographical thing. It wasn't something that was forcing them to go over there. It was just a behavioral thing. And because they went to those other areas of the lakes, they then basically developed into different species because they had to adapt to the different environments or the different sort of circumstance that was happening over there at the other side of the lake. So do you have any questions so far? Are you confused? Is everything okay?
Everything's perfect.
Okay, good. All right. So the role of mutations in speciation. So small genetic changes can contribute to reproductive isolation. If you remember, we said reproductive isolation is that mechanism where these animals are not able to reproduce together again, right? They once were able to do that as they were the same species, but then after speciation happened, after all of these genetic mutations happened and changes and everything, they are now not able to reproduce together again because either they're not able to form the zygote or the offspring that they form does not live for long or is not able to reproduce itself, right? So, um, what's it called? So these mutations, basically, of course, as we mentioned before, are things that happen over time, right? They happen over time, and it is believed that, of course, it's because of these mutations that we got lots of these different species. Now, chromosomal mutations is also an example of something that can lead to speciation. So, not just specifically gene mutations, but chromosomal mutations also. And the best example of a chromosomal mutation that led to speciation could be what we can see in humans. So, in humans, our second chromosome, or the chromosome number 2, it basically is a Robertsonian translocation. And this basically just means that, you know, when we have these different chromosomes, we have what were they called again? Metacentric chromosomes, submetacentric chromosomes, for example, acrocentric chromosomes, right? We have, as we can see, in the acrocentric chromosomes, we have two short arms and two long arms, right? In a Robertsonian translocation, what would happen is that we would get rid of these two short arms eventually, as these two long arms would fuse together. So that is a Robertsonian translocation, a fusing which would happen between the two long arms. This is something which is only seen in humans, and this differentiates us from other primates. So this basically could be one of the reasons, which would of course mean that we as humans, of course, we cannot reproduce with other primates. So we cannot reproduce with monkeys and apes. Okay, and this could be one of the reasons because we have this chromosome that has this Robertsonian translocation, whereas they don't. Okay, so the polyploidization, again, we mentioned, this is basically, poly means a lot. So polyploidization is the duplication of the entire genome, entire set of genomes, which is very common in plants and basically allows for these plants to develop beneficial features. Okay, so for example, they may develop sweeter tasting nectar, for example, for the bees, or brighter flowers or more flowers so that they can attract the bees, for example. Wheat is an example of this, is an ancient polyploid, and because of this, wheat is able to sort of withstand maybe harsher climates because of this polyploidization, which happened ages ago for it. Okay, and as we mentioned, of course, in animals, polyploidization isn't something that happens often, but as we know, it is something that started in the beginning of evolution and allowed us to form more complex vertebrates. So now macroevolution is something that is based on adaptive radiation. So, what is this? This is the rapid diversification of a species. So, this is the rapid sort of changes that happen within what's it called? A species that basically makes them different species. Okay, and this normally happens when their habitat or the area that they're living in is exploited. For example, the Hawaiian honey creepers. So what could have happened with them is that the Hawaiian honey creepers, of course, would live in Hawaii, and the areas that they're living in may have been exposed to deforestation, where, of course, they're living in the trees and everything, and it could be that their trees were sort of, what's it called, removed, right? And because of this, they had to go and find different habitats. And because they had to go and find different habitats, they also had to go and find different food sources. And because of this, their beaks changed in correspondence to what they were actually eating. Okay. And another example of this is, of course, in the Darwin's in Charles Darwin's theory of what's it called, of the finches, of the birds that we spoke about earlier, which we're going to talk about here in this slide too. But putting the birds aside for a second, we have two other terms that we have to learn also. So we've got parallel and convergent evolution. Okay. So, parallel evolution, this is basically the traits, the evolutionary traits, which happen in animals, or not just animals, but species that are closely related. So for example, of course, if we have two species that once shared like a common ancestor, that are living in the same habitat, they're going to develop the same sort of traits to deal with that same type of environment. So, that is parallel evolution. Convergent evolution is when distantly related species that may be living in different areas, okay, would develop similar evolutionary traits because of the environment that they are facing. And an example of convergent evolution is in these two cactuses. So we have the North American cactus and we have the South African cactus. And even though they're living in completely different areas of the world, they have similar evolutionary traits, okay? As they may be dealing with the same kind of climate, okay, over there. Even though these two species are they might be unrelated, or they may, they may be unrelated, or they may be distantly related, they are sharing these same evolutionary traits as they are going through the same sort of climate. Okay, so island evolution. So here is the example of Charles Darwin and the finches, or the birds that he was studying. So as we mentioned earlier, he basically realized that depending on where these birds were living, I mean, they were all part of the same species initially, okay? And then what happened was that they were then separated, okay, into these different Galapagos islands. And then the different islands that they were separated to had different things or different resources available to them, right? So they had to then adapt to that, and then their beaks basically changed over time to adapt to the type of meals, or to the type of diet that they would consume. Okay, for example, the large tree finch and the small tree finch, and also the woodpecker finch, they, as we can see, have more grasping beaks. Okay, and this is because they were eating insects. So, of course, these insects are quite small, they might be quite fast. So, they need these beaks which would adapt to that, which would easily allow them to grasp onto these insects. Whereas on the opposite side here, we have, of course, the small ground finch, the medium ground finch, and the large ground finch, which would have crushing beaks. They would have really strong beaks, which would allow them to break open the seeds in order to eat them, because the island that they were living in only had these seeds available. Okay, do you have any questions? We're at the end of this point. So, human evolution. Okay, so we first start off with the order primates. So, primates are the order of mammals that includes humans, the ancestors, and the closest relatives of these humans. And they first appeared in the fossil record around 85 to 65 million years ago. And this was in the Cretaceous period. You're not expected to know which period, like the names of these periods and dates and the years. These are not things that you're expected to know. As we mentioned before, you should have a overall general understanding of what it is that is going on. Okay, so these early primates, they were small, they were nocturnal, they were omnivorous, and they were living in an environment that was mainly dominated by dinosaurs. Okay, so they, what's it called? Their survival wasn't, of course, the best, but they sort of had to adapt to, of course, their environment. Okay. Now, because they were surrounded by these dinosaurs, they had to live in the trees. Okay. They were tree dwelling. So they had a tree dwelling lifestyle. They also had forward facing eyes in order for them to basically see around them better. For example, in order for them to be able to perceive depth. So, depth is like, for example, we have, of course, obviously, we have forward facing eyes also, and we have good depth perception, which basically allows us to go upstairs, for example. Okay, so they needed this to be able to climb up these trees and to see around them. They also developed opposable thumbs and long, flexible fingers and toes. And of course, this was to allow them to sort of grab onto the tree branches because, again, this is where they were living. They also developed short fingernails instead of claws in order to give them a better grip. Okay, now what was their social structure, or what is their social structure like? So, primates, excluding some nocturnal species, are highly social animals. An example of this would be chimpanzees. They basically learn through socialization, okay? And this basically just means that they learn through their mothers, okay? And usually, the primates or chimpanzees, for example, which are raised by a mother, have better social skills and are better able to sort of adapt to their environment. So they know where to go to get food, or when there is danger and things like that. They know this because they were taught this. Okay. So this here is also very important to know. So the ability to synthesize vitamin C. Okay, so we have two different groups of primates. So we have haplorins and we have strepsirrhins. Okay, so we belong to the first group. All right, we belong to the haplorins because we are not able to synthesize vitamin C. Now, what is the reason for this? So, there are certain things that our body needs, right? There are certain things that our body needs which we can either only get from our environment or which our body is able to make on its own. Okay? So, we mentioned before, for example, there were certain animals which lost their taste buds for certain things because they weren't eating those things, right? For example, we mentioned animals like cats, for example, they have more of a umami, they have a developed umami taste bud rather than the sweet taste bud because they're not really, they're not adapted to eating things like fruits and sweet tasting things. They're more adapted to eating things like meat, because they're carnivores. Anyways, so the haplorins, they lost their ability to synthesize vitamin C because over time, their body basically realized, okay, so these primates, they were living in an area where they were they had an abundant amount of fruit around them. Okay, so they may have had apples, they may have had oranges, they may have had a lot of these fruits that basically are rich in vitamin C or lemons or whatever, and because of this their body didn't need to make the vitamin C anymore. Whereas other animals which were in an environment where they didn't have these fruits available to them, they became their body became adapted to making that vitamin C, which would then of course allow them to survive. Now, Adnan, as you are in second year and you have your biochem, or you should have had your biochem, I'm not sure. It's either coming up or you're going to have it soon. Do you remember, or do you know what the importance is of vitamin C? Why is it important that our body gets vitamin C? What's the
So
purpose of vitamin C?
vitamin C has a lot of properties. One of them being our body uses it for the connective tissue synthesis. So
Exactly.
yeah, that's very important for our tissue, for the collagen synthesis, for our connective tissue. There's also
Hmm.
immune functions, antioxidant functions too.
Exactly. Exactly. Very good. So, Vitamin C, we need it in order to make collagen. As we know, collagen gives us our tensile strength, right, within our connective tissue. Vitamin C is also a electron donor. Okay, so when our body goes through oxidative stress, vitamin C can sort of combat this, right? So basically, it reverses that oxidative stress. Okay, so now the simians, they are animals like the monkeys and the apes, and they have large brains, reduced olfaction, meaning that their sense of smell may not be the best. They have vertical foreheads and reduced facial protrusion. They have diurnal activity, which basically means that they are active during the daytime and they sleep at night. They have color vision. Do you remember which photoreceptor was important for color vision? Which gave us color vision, actually?
Which photoreceptor?
Yeah.
Was it just the rods?
No, it is the cones. The
Okay.
cones are important for the color vision. The rods, they help with being able to see in the dark.
Yeah, yeah, yeah.
Okay. All right. And then the monkeys and apes also, of course, developed a complex vocal communication in order to communicate with each other, of course, and a simplex uterus. And that simplex uterus was the, what's it called? It was the doubling up of the oviduct. Okay. The doubling up of the Mullerian ducts. Okay. Now, this is not that important. Okay. Now, we're going to talk about, after the apes, actually, we have to talk about the evolution of the hominins. Okay, cool. So the apes is the next group that we have to talk about. So these apes include the gibbon, as you can see, which is the smaller one, the orangutans, the gorillas, the chimpanzees, and of course the humans. So this is the superfamily Hominidae. Okay, now they have a larger body size and brain. They eventually lost the tail. They have the ability to walk upright briefly. The menstrual cycle appeared within the apes, within the females, of course, and there was a division between the gibbons and the greater apes. Now we're going to talk about. Wait, yeah, sorry. So now, the next thing to know about the about not the apes, but specifically the group great apes, including humans, is that we have extended childhoods and high parental investment, which basically, of course, leads to better socialization, okay, and learned behaviors, and then, of course, better survival too. It also led to advanced tool use and cultural behaviors, high intelligence and problem solving abilities, all of which came from this extended childhood and the high parental investment. Okay. Now, with the hominin development, now you're not expected to know. The thing is, you can learn it this way. Okay, so you can learn, if you want to, you can learn only going from the afarensis to the Homo sapiens if you wanted to, but I have included all that are within the lecture slides. But what's most important for you to know is what came with each group of hominins, what came with them. But first of all, the hominins are a group that includes modern humans and their closest extinct relatives. This group split from the common ancestor with chimpanzees around 6 to 7 million years ago. Okay, so this picture basically shows you this. We have the hominin ancestor here at the bottom, and the split that it's talking about is right here. So it split between the chimpanzees and then the rest of the hominins, and then eventually, of course, the Homo sapiens. Okay. Now, most of these hominins lived in Africa, and most of their evolution happened within Africa, and then they eventually traveled outwards to to what is known as Europe now and the Middle East. Okay, so now we're going to start off with the first one, which is the ramidus, the Artipicius ramidus. Now, this, of course, as we mentioned before, most of the primates or all of the primates in the earlier times, they were forest dwellers or they were living in the trees. And of course, because this species was living in the trees, as we can see from these images, they would have long grasping feet and hands, right, in order to hold onto the tree branches. Okay, they also had a upright posture, and they had a small brain. They didn't have any canine dimorphism. Dimorphism is basically a difference in size between the female species and male species. An example of dimorphism is, for example, in that you have to learn about with parasitology. Okay, is in the arthropods, specifically in the black widow spider. So, as you may know, the black widow spider, the females are much larger than the males, and this is because the females only use the males for sexual reproduction in order to have their offspring to lay their eggs, and then they eat the males in order to use the males as basically nutrition for their eggs, in order to basically form their offspring. So that is an example of a dimorphism, where basically within a species there's a difference in size between the females and the males. Now, this was not existing in the Ramidus. In the Artipithecus ramidus, there was no dimorphism. Females and males were the same size. Okay, now the next one is the Afarensis. So these were living more so in the savannah, and at this stage they were fully adapted for bipedalism. What is bipedalism?
Are you asking me?
Yeah, what is bipedalism?
Bipedalism. Um,
What do you
is it
think it is? Word. Yeah.
um is
So
it
think of
the coordination basically? Basically, the ability to walk with the two legs, the
it.
coordination with the two
Exactly.
legs.
Exactly. Bipedalism, by means to pedals, I guess would be like using the two legs. But yes, bipedalism is being able to walk using two legs. So now this species, the Afarensis, were now able to actually move with their legs very well. Okay. At this stage, they still had a small brain. They had less teeth or smaller teeth, but they were very good at walking. Okay. And this is important to note. Okay, since they are eventually going to go from living in Africa to living in, Europe and the Middle East. And how did they do this? Well, they did this by walking, right? Okay, so the next one is the Homo habilis. Yes, Homo habilis. Now, they developed a reduced facial skull and teeth. They now start to develop a larger brain. Okay, and of course, since their brain became larger, this also meant that their skull became larger to protect the brain, right? And because their skull became larger and their brain became larger, of course, the pelvis also became larger. Okay, because they still need to, go through pregnancy and push out the babies. So the pelvis needs to be large for the infant's head. Okay. And at this stage, they also started to use simple tools, simple stone tools. Because, of course, you have to remember with a larger brain came more complex abilities, right? So, more complex cognitive skills, maybe. So, at this stage, they might have been able to think a bit more, and that's why they were now able to use their stone tools. Okay, then we have Homo erectus, and these actually are very, very, very important. And you may be asked, actually, if you have the syllabus point, you may be asked which of the hominin species were the first to migrate out of Africa. And this was the Homo erectus. Okay, so this is very important to remember. So the Homo erectus, they had a reduced facial skull and teeth. They, of course, developed a larger brain. Of course, because of the larger brain and the larger skull, they had a larger pelvis, and they lost a lot of the body hair, and they got darker skin. They were better at using their stone tools, or they developed their stone tools a lot better. They were now able to control fire because, of course, larger brain comes better thinking, or larger brain becomes better survival skills, right? And as we mentioned, they were the first hominin species to be able to move out of Africa. That was the Homo erectus. Then we have the Homo heidelbergensis. Now, these ones, they developed, of course, again, a larger brain. Their skeleton is the most similar to modern humans. And they, of course, had also moved out of Africa. And they're known as the first archaic humans, meaning that their tool use was very, 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 very good. Okay. Yes. Right, now we've got the next group. So the Neanderthals. Neanderthals. I can never say this word properly, but the Neanderthals. Yes. So we mentioned that the Homo erectus were the first to move out of Africa, and we mentioned that the next hominin species actually are, of course, they're moving out of Africa and they're going to walk to Europe, walking to the Middle East, etc. So the Homo neanderthals, they lived in Europe. They had a very, very large brain, they had a robust build, and they had this build which would allow them to adapt to the colder climates because, of course, in Europe, it would be colder than it was in Africa. Okay. Then we have the Denisovians. So these, we know a lot of our information about them from DNA evidence, and the DNA evidence basically says that they had a similar genetic makeup to Neanderthals. So they had dark skin, eyes, hair. They had larger molars. The molars in our mouth are responsible for what? Do you remember? Do you remember what the molars in our teeth are? The molars are teeth, but do you know what the molars are important for?
Yeah, sorry. So the molars basically they're large and what they do is basically chew and grind food.
Exactly, chew and grind food. So exactly. So the molars would basically help us with mastication, as you mentioned, and more or usually animals which have a lot of molars or more developed molars are animals which have more of a sort of herbivoric diet. So they're eating like things like greens and vegetables and plants and stuff like that, as well as omnivorous animals. So animals which are not only eating meat but also eating more plants too. Okay, so them having larger molars would then suggest that their mastication became better, and maybe also at this stage they were eating more plants too. Okay, so where would you find these Denisovians? You'd find them in Melanesia, so basically Middle Asia, Eastern Asia, Australia, around those areas, but all across Asia essentially. Now, the Homo sapiens, of course, their origin was from Africa. They moved out of Africa to different regions over the next 185,000 years. They have highly adaptable traits, or they are highly adaptable, have large brains and complex tools and culture. Okay, so what are the evolutionary milestones? What is it that you need to take away from human evolution today? You have to take away that bipedalism was one of the early traits which we developed, and this came from the Australopithecus species. Okay, they were the first species, or the Afarenus. They were the first species to be able to become very good walkers. Okay, and then the tool use. So the Homo habilis were the first species that were allowed that were able to use tools. Okay, and this became more advanced in the Homo erectus. The brain, as we mentioned, became larger and larger and larger as we went through each species. And of course, as we mentioned, the brain becoming larger basically contributed to them being able to use more tools and then being able to survive better, and then of course them being able to migrate out of Africa. Okay, so they migrated out of Africa to Europe, to Asia, to all these different places, possibly to sort of find other food sources, you know. Okay, and then there is some possibility that there may have been some interbreeding amongst these Homo sapiens, amongst these groups. For example, the Homo sapiens, the Neanderthals, and the Denisovians. Okay, because some of these groups actually lived around the same time as one another, and them basically interbreeding together would eventually contribute to the genetic diversity of humans that we see today. This is not that important. That's fine. We have our last point about the molecular mechanisms of evolution. So, for some reason, the lecture slides have included this, and this may seem kind of long-winded and confusing, but we're going to get through it together. So an analogy basically just means a short story. Okay, but first, before we get into the analogy, you have to know what a tinkerer is. So a tinkerer is a person that enjoys fixing or experimenting with machines and parts of these machines. So they're just fixing and experimenting with no clear goal in mind. Okay, so natural selection is compared to a tinkerer, is compared to a person that's just experimenting with no specific goal in mind, rather than an engineer, because an engineer is fixing things or trying to make things with a clear goal in mind. Okay, so evolution basically uses whatever is available to create functional outcomes rather than working towards a specific design or a specific goal. Okay, so that is what natural selection is. Okay, so what is molecular evolution? So this is evolution that happens at the scale of DNA, RNA, or proteins. So evolution that happens specifically within the DNA, the RNA, or the proteins. And this is normally information that we can get, or information that we need to be able to study molecular biology, evolutionary biology, and population genetics. Understanding how DNA, RNA, and proteins evolve has helped us with whole genome sequencing, protein characterization, and bioinformatics. So basically, allowed us to further understand how our genes work. So, what is the gene structure in eukaryotes? So, we discussed this a little earlier, where we spoke about how genes have, of course, introns and exons, right? And we mentioned that the exons are the coding regions, and meaning that they are going to code for a protein, and then the introns are the non-coding regions, okay? And instead, since they're not coding for anything, what they're going to do is they're going to just be there for regulation. Okay, their purpose is regulating the gene sequence. Okay, so we've got something called the synonymous and non-synonymous substitutions. So synonymous are substitutions or changes which happen within the gene structure that don't change the protein function at the end. Okay, then there are non-synonymous substitutions. These are changes which happen within the gene structure that actually do change the protein function. Okay, so protein evolution and gene recycling. So proteins that have the same shapes but have different functions are known to have, or are thought to have, a same or come from the same ancestral gene. Okay, so they have similar genes but different functions. This means they may have come from the same ancestral gene. Okay, as we mentioned before, this could the reason for why they might have different functions but the same shapes could be because of gene duplication, which happened earlier, right? So these genes could have been duplicated, modified, recombined, and this is why we have these new proteins that have the same shapes or similar shapes but have different functions. Now, conservation is something that's also very, very important to know when it comes to understanding genes and understanding DNA sequences. But basically, the way that it works is that if our body, or if our DNA essentially realizes that something is working for it, yeah, something is working for it, this is fine, this is perfect, it's not going to change that thing. Okay, so it's going to conserve it instead. If something is working for it, it will not change it to something else, it will keep it as it is. It will only change things if there's a problem. Okay, so the DNA sequences usually remain unchanged over time. Now, what are the processes that drive molecular evolution? Why does molecular evolution happen? Well, it will happen because of natural selection or genetic drift. As you mentioned before, natural selection happens, which will basically allow for a species to become advantageous in specific habitats or environments. Or genetic drift can happen, which are the random changes in the allele frequencies, which can happen because of things, for example, such as bottlenecking events. And this would lead to molecular evolution. We have something called junk DNA, and this is basically repetitive DNA sequences that don't seem to have a function, but are still within our genetic sequences, within our set of genes, within our genomes. Okay? So these repetitive DNA sequences, they don't seem to have a known function, we call them junk DNA. It can be possible that in some cases, these junk DNA may sort of develop some mutations which will allow them to become DNA that have functions, but it will never be that DNA that already have existing functions will ever become junk DNA that don't have a function. This is not something that ever happens. Okay? Examples of junk DNA can be found within the kangaroo rats. Okay. So, what are the gene duplication mechanisms? So, the mechanisms for a gene to basically duplicate itself. So, this is, for example, a whole genome duplication where we get the extra, of course, what's it called? The duplication of the chromosomes, and this leads to speciation. We have aneuploidy, which is basically just adding an extra chromosome, one extra chromosome, or missing a chromosome, and usually this is detrimental. This is not usually a positive thing. Then there is retrotransposition. This is forming retrogenes. These are genes that have been copied from a mRNA transcript and then basically made into sent back into the set of genes into the genome through reverse transcription. Basically, what it means is that these are genes that don't have introns. That's all that it is to it. These are genes that don't have introns. Uneven crossing over can also, of course, cause gene duplication. And as we know, crossing over is something that happens in prophase 1 in meiosis. And then replication slippage is basically when we are producing short genetic sequence duplications. And this is something that can happen randomly. Okay, so these are all examples of how genes become duplicated. You're not expected to know all 5, but of course, the more you remember, the better for you. If you can name two or three examples, then that's also okay. Okay, so the evolutionary implications of gene duplication. So, if a gene becomes duplicated, what will this mean? What can happen? Well, it can result in creating pseudogenes. These are pseudo basically means fake, so fake genes. These are genes that are non-functional. Okay, they are not going to make proteins which are working properly. Okay, or redundant copies. Again, proteins that are not working properly. Neofunctionalization is this will lead to these genes will basically code for proteins which will form new functions, and subfunctionalization are this basically means that these genes are going to create proteins that have divided functions. Okay, so they have either they might have a primary function, but then they might also have a secondary function too. Okay. Now, gene homology. So we have two things to talk about here. So two different types of genes. We have orthologs and paralogs. So orthologs are genes in different species that came from the same ancestral gene. And usually because of this, they have similar functions. And then paralogs are genes that basically are inside of the same organisms that happened as a result of gene duplication, and because of this they develop new functions. So if we look at this image and we make sense of it together, we have our ancestral gene right here. You can see the A with the 2 quotation marks. Okay, so gene duplication happened and we still see the same A, right? But on this side we only have 1 apostrophe, and here we have 1 set of quotation marks, right? This gave us 2 new organisms. We got the X organism and we got the Y organism. As we can see here, we have the A with the 1 apostrophe and the other A with the 1 apostrophe. So now, in regards to these two, what do they have in common? Well, they are both orthologs. Okay, they both have genes that they have a gene that came from the same ancestral gene. Okay, came from the same ancestral gene. So we can see we have the A with the apostrophe. Okay, and then we said paralogs are genes that are in the same organisms, but they happen as a result of duplication, and because of this, they have different functions. So, for example, this XA here. So this XA here and this XA here. So they are the same organism, okay? But they have two different genes, right? They've got the A with the apostrophe and then the A with the single set of quotation marks. This basically means that this organism and this organism are going to have new functions, whereas this organism and this organism are going to have similar functions. I hope that makes sense. Okay, so now gene conversion. So one allele is normally converted to another in DNA repair, and this can cause evolutionary changes. An example of this is the SIGLEC11 gene, which is found in humans. Okay, so this gene is important for basically controlling our immune response. So, our immune response, this is basically, of course, our body's reaction to, for example, a pathogen or an allergen invading and coming into our body. When this happens, our body starts to form an inflammatory response. But this SIGLEC11 allele, okay, what it does, or gene, I should say, sorry, the SIGLEC11 is a gene. Now, this gene is involved in anti-inflammatory and immunosuppressive signaling. So basically, it stops the body, right, from going into excessive inflammation, right? So it stops the body from doing too much, okay? Because if the body just continues to do what it wants to do when a pathogen has invaded, it will eventually lead to the body's self-destruction, as tissue damage will happen as a result of too much inflammation. Okay, so now, genomics is the last thing that we're touching on today with this syllabus point. So, genomics is basically the study of genes. Okay, so this is where the genes are being studied. We are sequencing the genes, we're putting them all together to study them, assembling them, analyzing them. And a good example of when something important was done with these genes before was in the Human Genome Project. So this is a project that lasted 13 years, started in 2003, ended around 2013 or 2010. I think it was 2010. Around then, basically. And basically, it was a lot of scientists that came together from all around the world that basically came down to sit together, or not sit together, but basically work in their individual labs to try to map out all the possible genes which you can find within the human DNA. Okay, and they were trying to do this so that they could label them, they could number them, and they can then sort of study them further to try to figure out, okay, in this genetical disease, which gene is it that actually, causes this disease, or what happens to this specific gene in this disease? Does it, become deleted? Does it become mutated? Like, what happens to this gene? So that's basically what they were trying to do in the Human Genome Project. They were doing genetic sequencing.